my appreciation is that engineers really try to apply what we understand to meet real-world needs. Biology had not really had that engineer's eye applied to it, but looking at the ability, for example, to put pieces of DNA together without it being an artistic endeavor, that, you know, it's not an ad hoc endeavor every time you want to put two pieces of DNA together. Now there can be protocols and standards that apply to all pieces of DNA that allow them to be assembled in a reliable way. Seems like a way that other engineering fields have scaled and become reliable, and had not really been tried really with biology before. So in coming to MIT as a scientist and teaching in a department that is an engineering department, I saw how effectively these engineering challenges could be used to engage students and have them learn the science as well as discover new things about science. I mean, that's sort of an interesting thing about synthetic biology, is that you have both the ability to build new things and have an output that you're hoping to see and generate, but also in the process of making that or trying to design it, you learn new things about the science and understand your limitations of scientific understanding at that point. And so I found it a very effective way for students to be learning the science and doing the really hard work of learning. And I was really just eager to share that kind of teaching and that kind of learning outside my walls of my MIT classroom. And so BioBuilder really came about in some ways because I felt like I outgrew my MIT classroom a little bit, and I wanted to bring this kind of content out into the broader, bigger community. BioBuilder starts with the materials I had been teaching with at MIT and have been teaching with, and ongoing challenges in the field. I mean, so for example, one of our activities starts with the hope that you can generate bacteria that will smell not so stinky, but actually smell pleasant, like bananas. And we ask the students to design a genetic program that will regulate that output of the cell so that it only smells pleasant during a particular phase of growth. And you might think it's kind of a silly project, a small sort of toy, but really scent industry is enormous, and the ability to control outputs of cells is key to really any biotechnology. And scent has been an underutilized reporter for cellular behaviors. And so in some ways this is a really important project, even though, you know, making cells smell like bananas may seem like a not earth-shattering kind of an endeavor. But the materials for carrying out that investigation are online at the BioBuilder site now, thanks to some teachers that I've worked with to craft them into teachable modules that fit into high school settings and college settings. And so students can start with animations that set up with those kinds of challenges are. There are hands-on laboratory kits that are provided through a distribution company that I've partnered with, so that students can then get into the lab with their teachers and study these questions directly. And then there's a third portion of the BioBuilder content, which is an online portal for students to share what they've learned. I think that that's something we do as scientists and engineers all the time. We have the opportunity to talk about what we've learned with each other. Students have a portal for sharing what they've discovered on the BioBuilder website as well.